Welcome to the Conversational Flow, a KCAS Bio podcast.
Hello, and welcome to the 32nd episode of the Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the weekly bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Hi, Adam. My name is Brian. I've been in the flow cytometry CRO space for over 14 years.
Brian and I are members of the growing leadership and advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas, with additional locations in Europe and a partner location in Australia, Brian and I are in Philadelphia, Jeremy's in Kansas, and so we are truly all over the map. We're ecstatic to have you listening to our 32nd episode of the podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you'll likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will feature a review of the latest news and resources and a focus on a topic of our choosing before discussing any feedback we've received from you. We're constantly on the lookout for topics, and we'd love to discuss something that you want us to cover. So again, we're elated to have you here and looking forward to a fun episode today. To kick us off for episode 32, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We've got a festive agenda today. And we'll, of course, start out, as usual, with our medical research news. And the first item on this section is Gilead's Trodelvy that got some additional FDA approval for TNBC, which is helping out with its overall antibody drug conjugate ADC process here.
Right. And so TNBC, we've got triple negative breast cancer there. So this is actually an expansion for first line treatment of unresectable or metastatic TNBC. And so what that means is that TNBC negative for estrogen receptors, progesterone receptors, and excess HER2 protein. This makes up 15% of all breast cancer cases and tends to grow and spread faster. So you have a worse prognosis. Now, what they're doing here is targeting TROP2, which is trophoblast cell protein antigen, cell surface antigen 2, which is overexpressed in a wide range of cancers and would indicate more of a poor prognosis and aggressive tumor growth. I did, in doing some research for this, find a paper, The Evolution of TROP2, Biological Insights and Clinical Applications in the European Journal of Medicinal Chemistry. This was published October of 2025. Really gave some insight as to the target here, why it has become important, starting to see more therapies there specifically. The thing I was really interested in were ADCs, right? So ADCs are not new. Antibody drug conjugates have been around for quite a long time, but they weren't as in favor as they are now. And so curious, Brian, how much background do you know on ADCs? Why are they becoming hot again?
So ADCs are wonderful because they are what's called a platform technology, right? You do all these safety studies on the antibody, you do all these safety studies on the payload, which is that active molecule you can tag onto an antibody. And then once you show that the ADC is safe in one case, you can sub out these payloads and really accelerate a lot of your clinical trial testing and get some interesting sort of differentiation and profiles in there. So it's very interesting from a drug development perspective that each molecule is its own sort of platform technology, even though it's all sort of from the same base antibody targeting.
Now, having come from Centocor and a true antibody company, right? Is interested to find out a lot of the evolution in the ADCs is actually the improved linkers, right? So the payload staying on the antibody is targeting it to where it needs to go, and then bringing this payload, to your point, on. But what would end up happening is these linkers may not have been as effective, and so there was more off-target toxicity. Now there's a better safety profile, to your point. There's better potency because it stays on, gets to where it has to go. The other thing is here, by moving this up in the treatment chain, it opens up to more patients, right? We're starting to see the first line therapies, whereas historically a lot of ADCs were later in the process, when you've already started to see more resistance mechanisms accumulate. And now also these ADCs are being combined with other therapies, so that you're starting to see the PD-L1s, those other therapies that can certainly help and really make this an effective treatment for people who have cancer.
Yeah, I think definitely the broad applicability is key to making these pharma companies very interested in it, because the wider the population, of course, the more of these treatments they can sell. So that is attractive from a financial viewpoint, and the more people that they can help as well from a overall patient assistance perspective. And then, as you say, the ability to combine these with multiple approaches. You think about basically a shotgun effect where previously we could only target maybe one mechanism at a time. If we now target 3 or 4, we can reduce the ways that a cancer could escape from one of these mechanisms by targeting multiple. So it'd be unlikely for them to spontaneously mutate in a way that will help them avoid 2 or 3 different mechanisms at the same time.
Well, and I think this also highlights, too, the evolution of therapies, because as I mentioned at the outset, ADCs are not brand new. They've been around for quite some time, but they've had to evolve to be able to gain that better profile and become a more useful therapy. And it just brings to mind the whole, I'm going to bring it up, we do every single podcast, chimeric antigen receptors, where for the longest time it was the hottest thing, and then it seemed like it was falling out of favor a little bit as a lot of modifications need to be made, and it's something where you just can't lose sight on these therapies. They just need the fine tuning. They need the experience, the knowledge to be able to get to where they are truly effective. And so it's kind of what I'm looking at here for the evolution of ADCs, and hopefully seeing that same path for chimeric antigen receptors.
Sure. No, I mean, if you look at the history of monoclonal antibodies, that was definitely a 50-year journey in itself. So if ADCs follow relatively close behind that, then they're sort of next up. Then, of course, we've talked before about bispecific and trispecific antibodies also on the docket. And then, yes, as you say, chimeric antigen receptors definitely next on that list. But, in terms of mechanisms, maybe it's a good time to segue into that second paper, which is a retraction, actually, for a PD-1 study about lung cancer survival related to timing of treatment.
And I saw you did there, a good time to transition. Man, you're getting really good with these transitions. So Nature retracted a PD-1 study that tied lung cancer survival to treatment of the timing. Now, what they get into is circadian rhythms, right? And I've been interested in circadian rhythms for a long time. This is just how your body changes over the course of a day, the rhythms of your body, responding to day and night. Obviously, we go to sleep during the night, we wake up during the day, or certain aspects of our body function different as we go throughout the course of a day. And so there has been theory out there that the timing that you administer a treatment can matter and amplify or hamper the therapy itself. And so this paper came out and it basically said exactly that. Oh, the huge impact. But that was the problem. Looking at this, it wasn't that there was an impact. It was the magnitude, right? And so there was a lot of stuff that was not taken into account, the confounding variables, excuse me. And so after question, it got pulled back. I will say, though, I don't anticipate this is the last time we see this. We're certainly going to see more studies as to the timing of it. I mean, we've got tons of data as to the administration of therapies and impact. And I think with AI and the ability to mine that more effectively, we're going to start to see and be able to parse out what the impact is. So while yes, the paper was retracted, I think that it still highlights an area that is key for study.
I agree. I mean, there's a clear mechanism there, right? Where, as you say, circadian rhythm does regulate the presence and quantification of multiple different types of hormones. So it's easy to see how that could have a knock-on impact to multiple different kinds of therapies with the activation status of different markers in relation to those. So I would agree there would be more studies. The one thing I might add, though, is that I think that these would be investigator-led or maybe hospital-led studies, as opposed necessarily to pharmaceutical studies, just because you can't commercialize a drug based on treating earlier. Maybe they would, modify their dosage regimen during the trial for that, but I don't think that they would dedicate an entire clinical trial just for that purpose.
Well, that was part of the reason why this was retracted. When I'm talking about the magnitude of the response, it was nearly as effective as the treatment itself.
Yeah.
And that's not what you would expect. And to this, when people question, well, why would circadian rhythm really be impactful? You're talking about just as simple as cortisol levels, right? At certain points in the day, your cortisol is higher, you have more stress, and at other points, you have lower cortisol. And that can dampen responses or alter responses of your immune system. And it has been shown that things like vaccines can see a variation in impact just based on the timing of the day, the impact administered, excuse me. Not obviously this magnitude, but that there is some difference there. So, and it would be great too. This is sort of thing that you can follow by flow. Right? You do a study here and you have your flow data. You can look at the difference in activated T cell levels, the NK cell levels, checkpoint inhibitor expression, T cell proliferation markers, these sorts of things. I mean, really, flow cytometry lends itself really well to studying.
Yeah. I just hope that they find that everything is more effective when you do things in the morning, because if they say you have to wait until like 8 p.m., then that's going to just make all of our lives a lot more difficult. So,
well, it did say this study was before 3 p.m. So, yeah. I mean, it sounds like it's still going the same direction that you want.
Right. Goodness. Okay. So then just to sort of flip over to our next story with no transition whatsoever, I'll just make the abrupt switch. Switching over to investment news, Insight has made a deal with Star Therapeutics to take over a late phase constellation. So this news article took some literary license there. But it's for a total of up to $2 billion, depending on if we hit milestones or not.
Well, and, it's from Star, Star Sure. Therapeutics. It's a subsidiary, Vega Therapeutics. And so they're primarily centered around VGA-039, so phase 3 monoclonal antibody. This is against targeting von Willebrand disease. So it's a factor for platelet adhesion. And so trying to essentially rebalance coagulation, and has shown promise, reducing bleeding rates, and the convenience is it's a monthly administration, sub-Q. And so the tagline here, it represents essentially Insight's largest business development transaction under the CEO, Bill Murray, and designed to diversify revenue. So, a lot of the big pharmaceutical companies have patent expiration, patent cliff, right? And at some point, you can't depend on the revenue for some of these blockbusters. They go into generic and there's a lot more competition. And so what they try to do is keep that pipeline moving, get other assets that they can then leverage to maintain that revenue.
That's it. And definitely more and more pharma companies are purchasing these assets at phase 2, phase 3 levels to make sure that it is fully de-risked and they have a clear plan on what to do with their pipeline. So they'll pay a premium for later stage assets, but they'll be more sure that they can address these pipeline gaps whenever they are nearing their patent cliff.
Right. Yeah.
Okay. And then for the next investment news, that this hits a little bit closer to home for us here in the lab, where Merck is purchasing Biotechne, and it's a total of an $11.3 billion deal, which is huge. They're comparing it to the 2015 purchase of Sigma, also by Merck.
Yeah, so they're consolidating life science businesses. Of any company, really, this is along Merck's DNA, right? As you pointed out, the Sigma Aldrich purchase, and they've got this side of their business as well as the pharmaceutical side of the business. And just really expanding offerings there. I personally thought this was really interesting, just having been in the lab in the past and purchasing reagents from Biotechne. Obviously, the Sigma has always been a big one, and seeing it consolidating like this. I'm curious if you see this as purely something for a company like a Merck, or do you see other pharmaceutical companies following suit on reagent company purchases like this?
I mean, Merck has been a chemical supplier for a long time, and that is the arm of Merck that we're really talking about here.
Right.
Less so about the pharmaceutical manufacturing, more so about the the raw material manufacturing. So they'll manufacture media and water and specific chemicals and starting points, so APIs, if you will, for pharmaceutical companies. So that's been a large part of their business for a long time.
Yeah.
So I don't think that would expand into other pharma companies. Like that's not a core part of their business. It's because Merck has always been a chemical synthesis company in terms of why it's doing this.
Mm-hmm. Yeah.
In terms of consolidation within the reagent supplier market, I do think that is happening pretty aggressively now. You can think about the reagent supplier market as sort of trailing the biotech M&A in general, and especially with the last 2 years that were sort of down cycle, you expect to see like contraction and consolidation overall.
Mm-hmm.
So that may have just trickled down to the supplier market at this point, with a post-COVID slump that allowed a lot of them to basically think about how do we inorganically expand our business if we're not growing organically.
Now, I definitely agree with you. I understand the idea of the two arms of Merck. However, having something like this, does this reduce the costs for enabling drug discovery and manufacturing for a company like Merck, because they have built up this sort of infrastructure that many other companies would not have?
No, the way the companies are structured, they operate sort of at arm's length. So I don't think that Merck KGaA gives Merck Sharpdone or any of the other Merck entities preferential pricing or treatment because they both have the name Merck in them.
Mm-hmm.
If that makes sense. So
It's interesting.
yeah, and it's the same way with Roche, right? Like Roche also has a pretty large diagnostic arm, but they don't give like special deals or whatnot to the parent Roche holding company, to my knowledge at least, due to the fact that they are sort of related. Everything sort of operates as independent businesses,
Mm-hmm.
which is how it is approved by regulators. Otherwise, it would be a severe concern, and pharma companies would not want to buy from suppliers that are linked to other pharmaceutical companies.
It's interesting. The reason I asked is I just really hadn't looked into that aspect, the business aspect of it. I'm not an MBA, so, really appreciate your perspective there.
Yeah, and you can think about it like from the perspective of Thermo too, right? Right, like Thermo owns a number of different. Thermo Fisher owns a number of different businesses. Some of them produce reagents. Some of them produce instruments. One of them is a CRO, right? And of course, the CRO is a direct competitor to KCAS. But we still buy their reagents because they're good reagents, and they don't give us like different deals than my understanding of CRO Thermo's deals are. So it's not really a conflict of interest from that standpoint. But I do think that this story speaks to the consolidation of the life sciences market. Basically, all the groups that are producing reagents are getting bigger and bigger, which gives them sort of additional control over their pricing strategy and likely increases the prices of reagents overall.
Right, right. No, it's interesting. So appreciate the information there.
Sure.
So moving along then, what do we got as far as information on KCAS?
Let's see. For KCAS news this time, we have solidified our fall and winter conference schedule. I think we're going to go to, of course, the MAFCA Annual Research Day on September 17th. Super excited about that.
Yes.
But then also the ASH conference in December. So that's the American Society of Hematology. So from a flow perspective, always exciting things to go see.
I heard rumors that you're going to be presenting at MAFCA. Is this true?
I will be introducing and hosting MAFCA, but I will not be presenting there. Definitely a few of our scientists here at KCAS will be there. Definitely with posters, potentially with talks, and a few, many researchers actually from the Philadelphia area have submitted their abstracts as well. So it should be a really engaging conference. We'll have both the current and future president of ISAC there. ISAC is the International Society for the Advancement of Cytometry. So it's the main group that governs flow cytometry. So both of the presidents of that group will be able to show up, which is great because, of course, MAFCA is ISAC affiliated.
Mm-hmm. Yeah. Now, I will say, too, though, your mention of planning for the fall and the winter just almost seems blasphemous. I understand why we have to talk about that now and plan. But I mean, it's over 100 degrees out right now.
That is one of the reasons I'm talking about it. That way I can remember it will not always be this hot. It will at some point be cold again. And we can all look forward to that time.
Mr. Hates Snow says it's going to be cold again. I love it.
Cold, but not snowy. That's what I'm aiming for. Absolutely.
All right. So what we got next? We have the paper. So you want to give your thoughts? I'll kick it off. This podcast paper is a survey on core flow cytometry facilities, instrument maintenance, usage, funding, and this was published in the Journal of Biomolecular Techniques 2023 by Sonal Priya, and I felt this was interesting just because it's a little bit of a different direction for us. We've talked a lot about data. We've talked a lot about clinical studies and study setup, validations, types of assays, the difference of instrumentation, but really not from the focus of, maintenance, usage, things of that nature. So what struck you about this paper?
As you said, I find this really interesting because a lot of these discussions and negotiations are always had behind closed doors. So to have a whole paper on it was interesting. Like sometimes I'll see discussion sessions at Cyto or other things about this topic, but I don't think I've ever seen a paper about it. But it's quite useful, right? Just to give people benchmarks. Am I paying too much to maintain the cytometer? Based on the age of the cytometer, what decisions should I make? So I found it quite useful there. Interesting to see how core facilities view this as well in terms of the ongoing maintenance of it and what they can do sort of in-house versus contracted.
I have to go with something funny first. So you have to allow me to indulge here. It was a line in the paper. Today, fluorescent antibody panels containing 15 or more antibodies are routinely used to identify unique cell populations with very distinct properties and functions. Now, what of that would I have found funny? Stumped you on that one.
I was about to say upwards of 15. Like
Yes,
what?
yes.
Yeah. Okay.
We're
Yeah.
talking about 15 or more antibodies. And yes, many of the clinical assays and whatnot, typically around that 15. But just with the amount of requests that we get for the 35, 40, 45 color, it really was thinking about the days of the 15 color panel, 18 color panel really stretching. And that's when we were on the Fortessa. And this paper goes into a good deal of the age of the cytometers, the cost for running, and interestingly, talking about how these older cytometers are still getting the job done. We've got a couple of Fortessas here, a couple of Fortessas out in Lyon site, and fantastic job. They are, the percent of days of service is incredibly high. And the cost to keep them maintained is lower than the newer instruments, primarily because, newer instruments, there's newer technology, costs more to keep it going. Reagents also are more expensive generally for the newer technology. And so here, I do want to take a step back. Where does all this information come from? You touched on it a bit, but this was 146 flow cytometry cores across 14 countries that they had queried and went through all the different topics that you had essentially pointed out. I think another thing I found really interesting for this is the staffing, right? The average core, you typically had two PhDs, 3 technicians, and maybe 1 or 2 additional staff. Felt like, for what they do, it's incredible. I mean, just cranking out work, it seems very lean there. What are your thoughts?
Yeah. I have, I guess, 2 big things. One, if we think about 90% uptime over the course of a year, that means, let's say that it's, call it 250 days in a year. That means it's down 10% of those days. So 25 days. So it's out for 1 month out of the year.
It's a lot.
That's a lot. So 90% uptime as a threshold is
Very low.
from a CRO perspective, like too low, right? Like it's, as long as you've got multiple instruments, maybe it's okay for a core, but from a CRO perspective, we expect, upwards of 99, definitely upwards of 98%, but probably closer to 99% uptime.
Yeah.
So that's always a consideration. And I would say, in terms both of your comment on the 15 color versus the 30 and 40 color and the size of the cores, I feel like with academic cores, it's sort of a tale of haves and have-nots, if you will, where if you survey 146 different cores, on average, they're going to be small like that.
Yeah.
And some of them are even smaller, right? Like probably 2 people, one person in some cases. But then there are some cores that are like 30 to 50. I think technically the university system of Belgium is unified across 17 universities. So then their core is actually, well over, I believe, 40 people, if I remember correctly. So they can be quite large too. And I think that's the delineation where the smaller cores are probably still on the 6 to 20 color assays, whereas the larger cores are all the ones that are probably doing the 40 to 50 color assays.
And we're talking about shared research labs, right? So where a lot of people may be using these instruments, and they talked about too, who are the people using them? Now, for the analyzers, users and staff, the thing that I really enjoyed was, like, my perspective of doing sorting in the past is it's staff driven, right? Nobody wants inexperienced users operating a cell sorter because they're so finicky to start. Even the best sorters, there's still stuff that you have to do to get them running, and you got to make sure the streams are lined, drop delay, you got to do, make sure there's no fanning, and they can be temperamental, and you have somebody on there who messed around with your settings. The next time you go on, all of a sudden it's a nightmare. So I was always amazed that I've heard stories of people who run those sorts of sorting cores and allow people to, do a training and then get on the machine. I think that would give me a heart attack.
Maybe 2 trainings, 3 trainings. I don't know, but yeah, certainly more than once would be needed to make me feel comfortable. That's for sure.
Mm-hmm. Yeah.
But analyzers, I feel like most cores will let users log into after one use. Those are usually a little bit more robust, especially with the Fortessas and some of the fax calibers and things like that have been running for 15 years. If they haven't broken yet, they're not going to break down now. So,
and did you see the average age of the analyzers? 9 years old. So these analyzers, they're getting the job done. They got most of the 9-year-olds are probably going to be in the conventional analyzers rather than spectral, but something interesting there.
Yeah, I mean, when you look at the stats on the repair costs, then you can sort of see why, right? And I think that goes back to the common phrase in Flow, which is to call everything a box, right? Mm-hmm. And it's just the Fortessa box. And then everything inside there over the course of 9 years has probably been replaced. It probably has new lasers. It probably has a new DAC board. Probably has a new high throughput sampler. So if everything inside the instrument was replaced over the course of 9 years, it's sort of like if I came in and had a robotic kidney and then I had a robotic heart, and then at some point all of my parts were new and robot. At what point is it now a new instrument, even though it's 9 years old?
So you're saying that when you're 80 and you got the bionic hip, you're really not 80. Yeah, I mean, the same thing goes through here. Now, how do you prevent it from getting to that point, though? They talked about preventative maintenance. And the one thing that's almost universal was having daily QC, monthly deep cleaning, preventative maintenance twice yearly. These are the things that are our hallmark, right? We've got SOPs in place, make sure that these kind of cleanings continue to happen. We want to have eyes on, are our machines passing QC every day? And if they don't pass, what's going on? What is the reason behind that, and how do we fix it? You're looking for the percent CV. You got to make sure that there's no clogging. Get the background down. Make sure that the instrument sensitivity is where it needs to be. And so I shouldn't say pleasantly surprised. It kind of, to me, was the no-brainer, right, of the paper is if you want these machines to last 9 years, 10 years or more, then you really got to be maintaining it well.
Yeah, I mean, from a core perspective, it seems like daily QC, maintenance, and user training are sort of the 3 key competencies, and everything else becomes a sort of a nice to have, right? So make sure that the instruments are in good shape and that they are performing according to some sort of specification, and that people know how to use them. And then everything else is on the users at some point.
Now, to your point, though, they did point out that the percent uptime for user-operated analyzers was at 92.5%. Then analyzers operated by both user and staff was at 87.9%. So, you joke around about 1, 2, 3 trainings, but it shows that when you add...
Sorry, Adam, you cut out a little bit. You joke around about what?
Well, you joke around about whether it's 1, 2, 3 trainings, right? But it really matters because you add in a group who may not be as trained as effectively, and you see almost a 6% drop or 5% drop in the uptime of the instrument.
Yeah, I mean, you can easily break things pretty quickly, especially with sorters. So, yeah.
Yeah. So what do you think? How do you tie this back to us? What does this mean for a CRO here?
I mean, this is why Natalie doesn't let me touch a SciTech. So I think that's point number 1. She's made reasonable decisions along that front. So that's always good.
You can't blame her.
Right. No, but I think, as you say, the commonalities there around the mandatory runs of QC to make sure that we have good perspective on how our instruments are performing, doing maintenance before it's required to make sure we can maintain that 98 to 99% uptime, and just the user training as a critical piece to make sure that instruments are functioning, let alone like the assays performing well, but the instruments just being available for use is critical as well.
Yeah. Yeah, I would actually offer our uptime is over 99%. The support team does a fantastic job of maintaining things and getting the appropriate people out to maintain the machine if we ever need to have a call. And they did go into this paper whether or not you have OEM service or you have your own engineer do it. Very multiple different ways on how to keep things going, and obviously cost is one of those things that is a consideration.
Absolutely. The one other thing I did want to call out from this was I did not realize that mass cytometers were that much more expensive to maintain.
Yeah, I did see that. I wasn't going to dive much into the mass cytometers just because it's not our focus. But you're right. I mean, you're talking what from 12,000 for your analyzer to 70 plus thousand for the mass cytometer.
Yeah, I mean that's massive. I mean, I understand with the detection methodology, like you have to pull rails and do things like that more often, but I didn't realize it would lead to that much of a disparity. So that was a cool thing to see in the paper as well. But yeah, great paper, and very useful for the field, especially, of course, in core labs, but also good to reflect on from a CRO perspective. So,
And
yeah.
these authors did have a couple of other papers like this. I didn't really get to dive into it. I just saw the titles of similar papers. And so certainly if you enjoyed this paper, you can look for other ones by the authors.
Okay, a whole series. Wonderful. Thank you.
Mm-hmm.
All
All
right.
right. So what we got next?
Yeah. Next up is banter, and it. Oh no, sorry. Next up is fan feedback, and I think related to this paper, best feedback question is: how far have you gone on repairing your own cytometer?
Does that also dovetail? Do you know when you've gone too far?
I think that's when the fax jazz shocks you a little bit. That's how you know. So
Well, that has happened. I mean, you've got the oh my goodness, the arms, the sorting arms, I'm blanking on the name, but remember, you could easily get arcing on those things, so you got to be careful. But it's funny, that wasn't the picture I had in my head. People doing their own repairs, and you got buffer leaking all over the place, and the panels opened, and yeah, it's craziness. So
I always get nervous at the point where there are those large stickers saying that if you do this, you will invalidate the warranty. That's always my point where I'm like, I don't know if I'm ready for this one yet.
isn't that the same though as if you rip this tag off your mattress, it's just punishable by a fine or imprisonment?
Yeah, and that's why the tags on all my mattresses are down there. That way, the mattress police don't find it and come get me, right? So,
For sure. All right, so banter. You pointed out. What is your favorite summer cool off treat? Perfectly applicable to it being ridiculously hot, over 100 degrees. I know I'm going to get into my car and my air freshener's got that little oil hanging from it. It's going to be bubbling or something. I will tell you, my favorite is I love getting a slushie. It used to be when I would do long runs in this kind of heat to come back and have a slushie. Oh, you could feel that cool going down. And I would always get a combination of the Coke and the cherry flavor. So you get your cherry Coke slushie. How about you?
that is refreshing. I would go with a milkshake, and that's always a little bit dangerous with dairy, of course, just like a whole shot of dairy and sugar all in one. But there was a place in Atlanta, and it's closed down now, but it used to take a whole red velvet cupcake and blend it up inside of a milkshake. So you drink the milkshake and also be eating a cupcake at the same time. This was just wonderful.
That sounds amazing.
Right? Should not do it frequently, but when I did it, it was wonderful.
I think that would speak to you'd be in different health if you did it frequently, wouldn't you?
That's true. Yeah, that'd be a concern.
So, what do you got planned for the 4th of July weekend?
So I'm actually taking all of next week off and going to go out on a family vacation and hopefully see some sea otters and whales and things like that out near Monterey Bay.
Oh, that sounds fantastic. Now, I think when you told me about the sea otters, you're going to yell at them. They misbehave? Is there a reason why?
Apparently, they're either mischievous. Barbara told me that they're vicious, which I don't know if I believe her or not. I think she's trying to scare me.
They look so cute.
But right, they're like little like puppies out there in the water, and they just always drift out there holding hands. I mean, how cute? How could you get cuter than that? So no, I don't actually intend to yell at them. I'll be polite and respectful at an appropriate wildlife distance. But I do enjoy seeing all sorts of wildlife. And that should be fun. So, and my kids will enjoy it quite a bit as well.
That's cool. For me, I'm going to be doing lots of Top Golf. So I ended up getting this Top Golf summer pass. Getting out there with my father again, going to bring my daughter, some friends. And so really looking forward to doing that. It was the first time in my life I actually expect to hit the ball when I swing the club. It's a novel thing for me. So, and my dad, who's been a golfer his entire life, he didn't seem too displeased with my progress. So I'll take that as a win.
Is that because you were beating him in Angry Birds, or is that because he thought your swing actually looked good?
So he did win most of it. I will say the last one that we played, he won most of them. But I was competitive. I did win 2 of the games. And so, it was more about the swing. So, and we didn't do Angry Birds. We were doing Block Party for the most part.
Okay, fair. All right. Well, I know Kansas City has a big golf tournament that they go to and send people to every year. So maybe if we get good enough here at Philly, we'll have to send a contender over there.
That sounds good. I like it.
All right. Excellent. Well, thank you to all of our fans for listening in. Our next episode will be coming out in just a few short months. So looking forward to having everybody join us again there.
Yeah. Thank you so much for listening. Really appreciate it.